Welcome, everybody. I'm very happy to be here opening this session. I mean, I was invited by my friends Milton and Shuba to somehow to work with them on the ONWARD webinar series. We are very proud to be supporting this webinar series and the ONWARD project. I mean, ONWARD network. Milton will say something more about the ONWARD network, and also you say something about the speaker, James Shutler. So, I mean, with no further ado, Milton, you know, have a nice, have a nice event.
Thank you, José, Luis. Hello, everyone. Good morning, good afternoon, or good night, depending on where you are. Initially, I'd like to thank José Luis Moutinho, Catarina, and the entire Air Center team, and everyone who is directly or indirectly helping us in this collaboration between the Air Center and the ONWARD network. The Open Network for Water-Related Diseases Network was recently created as a project funded by the Global Challenges Research Fund of UK, and is dedicated to developing a network of scientists and stakeholders sharing a common interest in better understanding of environmental controls on outbreaks of water-related diseases. The role of societal engagement in countering it, and in forecasting, early warning, and risk mapping of water-associated diseases through the use of remote sensing, field observations, and mathematical modeling. Our vision is to enable cost-effective, regularly updated, geo-referenced early warning for areas vulnerable to water-associated diseases, which in turn will enable preventive measures to be deployed in a timely manner to minimize the probability of epidemics. ONWARD is an open network committed to promote better dialogue and collaboration among experts from all relevant disciplines, including but not limited to microbiologists, molecular biologists, environmental scientists, remote sensing scientists, medical practitioners, and social scientists, and with stakeholders, including the general public. The project was initiated by Trevor Platt from the Plymouth Marine Laboratory and me, Milton Campbell, from the Brazilian National Institute of Space Research, Brazil, supported by Shubha Satyendranath for PML. Nandini Menon from the Nansen Environmental Research Center in India is also a partner in the project. The board of directors of the network, other than the three of us, are Dr. Grinson George, principal scientist from the Central Marine Fisheries Research Institute in India, Dr. Anas Abdulaziz, principal scientist from the National Institute of Oceanography in India, Dr. Bess Ward, faculty and chair, Department of Geoscience and the High Meadows Environmental Institute at Princeton University, Dr. Rita Caldwell, Distinguished University Professor, University of Maryland Institute for Advanced Computer Studies, Dr. Craig Baker Austin, Principal Scientist from the Center for Environment, Fisheries and Aquaculture Science in UK, and Dr. Nicholas Thompson, Professor, Wellcome Trust Sanger Institute, also UK. We hope you all join ONWARD and help us make it a success. If you are willing, please write to onward.gcrf@gmail.com with ONWARD membership in the subject line. We need to make the membership to grow, please. Now, following this introduction about ONWARD, I'd like to introduce you Dr. Jamie Shutler from the University of Exeter, UK, who will present and discuss rapid assessment of SARS-CoV-2 transmission risk for fecally contaminated river water. This is the fourth webinar of the ONWARD series in water-related diseases. There are links to environmental quality, monitoring methods, and solutions. Dr. Jamie Shutler is Associate Professor at the University of Exeter in UK. He's an ocean and atmospheric scientist who uses satellite Earth observations, in situ observations, and models to study and monitor land, water, and atmosphere environments and interactions, particularly in relation to climate. His work includes studying atmosphere-ocean exchange of climatically important gases, carbon accounting, bacterial, biological, viral, and chemical water quality, and land-water continuum interactions. In addition, he's applied his novel ideas to design monitoring methods for aquaculture, for global analysis of carbon, and design of satellites for ESA. A multifaceted scientific personality, his published research includes developing and exploiting computer vision techniques for biometrics, human gait, medical imaging, breast cancer, and surface ocean currents. He was an invited scientific reviewer for IPCC on special report on the oceans and cryosphere in a changing climate, and he was a lead author in UNESCO and IOC decadal vision for integrated ocean carbon research. Along with his international research programs, he has taken care to disseminate his research outcomes to the public through the Guardian Environment, BBC News, Al Jazeera, Forbes, Higgs, the Daily Mail, and the World Economic Forum. Jamie will make his presentation in English. Dear Jamie, I publicly thank you for your willingness to be collaborating here with us today. The floor is yours.
Brilliant. Thank you, Milton, for that very kind introduction. So, like I imagine everyone in early 2020, I found myself very much stuck at home, unable to travel anywhere, and juggling the complexities of limited access to everything, as well as the ability to homeschool my young daughter and continue in some way to work. And so this seminar is really a story about some of the things that we did and work related with my colleagues during this time. At the time, I was part of an international training network focusing on water quality. So this was a network that was funded across the European Union. There was about 20 different partners. And we were focused on coming up with and developing new and novel ways of monitoring water quality and developing sort of very small, low environmental impact sensor systems and deployment techniques. So these are sort of very almost disposable sensors, very tiny sensors, as well as deployable using drones and these novel kind of approaches. And this network was led by Ravinder, who's an expert in sensors design and manufacture and fabrication. And one of the other PIs that was involved, along with myself, was Christoph. He's an engineer and a mathematician. And so at the time, we all found ourselves in the same scenario. We were at home, but we were watching the news, and we also collectively had this, we felt compelled that we needed to do something to help, that we couldn't just sit back and watch what was unfolding. We actually had to actively be engaged in some way. And at the same time, in another part of my research group was Tom. Tom was he studied carbon, and so he was only focused on carbon. He wasn't focusing or working within this network. But he's a computer scientist and a mathematician. And his PhD was actually in modeling of malaria. So he had a background in epidemiology. And this he was a key member of the team that did this work. And the reason, and kind of I'll come back to the reason why I kind of highlighted this towards the end of my talk. So around this time, Christoph actually came across this publication, and this was really what started this whole journey for us. And this was a piece of work which was the first method where they'd used wastewater from a catchment wastewater, so it was a combined sewer and water usage. And they used the viral counts that they found within the sewer system to then estimate how many people within that catchment were actually infected with Sars-Cov-2. So this is the severe acute respiratory syndrome, which first appeared in 2020. And this was the first time this that people had done this. It was a method which had been used for other types of, they refer to it as wastewater epidemiology. It was the first time that it had been tested and tried and verified in some way for SARS-CoV-2. Now, this posed an issue because it becomes a potential fecal-oral transmission. Because if you have a viable virus within the sewage system, then anyone who comes into contact with or is in the presence of that sewage has a potential for the virus being transferred to them. And at first, I kind of thought you may think this kind of issue is only restricted to certain parts of the world, places where maybe the water sanitation isn't quite so advanced, or under certain circumstances where a sanitization system has failed in some sense. but it actually is a much bigger issue than that. So this is a news article, again, during the first year of the pandemic. This is in June 2020, and this is talking about a large sewage system failure where raw sewage is appearing in what appears to be or would normally be a tourism beach, and it's pouring out into the community areas. This wasn't unique. There are regular spills in the United States, in Georgia, Florida, and New York, as well as this one in Spain, all during the same period. And in fact, it happens in the UK quite routinely. So under European law, water companies can legally expel raw sewage into water systems under particular circumstances. So it might be that their system has become overwhelmed by volume, or it may be that they have an accidental failure. But it can and does occur regularly. And so this immediately for us, we were discussing this over email, and so this immediately comes up with this issue of, if you're getting sewage into the water system, whether that be in a coastal environment or a river, that means there's a transmission risk for the people using those water systems. So this might be surfers or kayakers, as you can see here, or people swimming in rivers. And this river swimming is something, particularly in the UK, which has become very popular during the first wave of the pandemic, because people were trying to seek and find a way of exercising outside with limited restrictions. But then we realized that this is a much bigger issue than just people wanting to use recreational spaces. There are large parts of the world where you have large communities which are living alongside large water bodies and reliant on those water bodies, where the methods of sanitization and sort of cleaning of the water may not be as advanced as other countries. This is Sao Paulo. This is a reservoir in Brazil, where you have a large favela, so a shanty town right next to the reservoir itself, where you may have reduced water sanitization methods being used, but also a very large population, which is highly reliant upon that water system, which is right next to them. And so the issue of this fecal to oral route is an international problem. And so, this was the point where we felt that we really needed, and we should do something to try and characterize and evaluate the risk, really, that this kind of transmission route posed. The next thing that kind of occurred to us is, if you're thinking about the human environment, what about the animals which are living in this natural environment as well? So the SARS-CoV-2, the way it's considered to be transmitted is via this ACE2 protein, which is prevalent in the human lung system, which is why we're susceptible to this. This ACE2 receptor, this protein receptor, is also in lots of other animals as well. It's in other parts of your body, the human body, apart from your lungs, but there's a lot of them in your lungs, but it's also prevalent in lots of other animals. And there have been cases in the literature. There's particular coronavirus, the family of viruses, coronaviruses. There is one that was identified as only existing within beluga whales, for example. And there's also a strain, cetacean coronavirus, which is particular to dolphins. So we knew that this is an issue to animals as well. And there was this work by Damas and colleagues that is now published in Proceedings of the National Academy of Sciences, but at the time it was available on a preprint server. And they categorized all the animals, common animals or animals they considered to be important, based on their susceptibility to the virus, which is linked to this ACE2 protein. So this is their table of all the highly susceptible animals. And if you look down the list, all the ones in reds are whales. So there's 6 different whales that are potentially are highly susceptible to the virus, and also dolphins as well. And so now we're at the point where we're thinking, well, clearly this is an issue for humans and animals that are interacting with or living within water systems, riverine and coastal environments. So, of course, we're then stuck with a problem, realistically, how could a whale actually become infected or a dolphin become infected with the virus? And the hypothesis we came back with was common to the initial issue we were looking at, the sewage spillage. And this is a news article again during the first wave of the pandemic, where this is talking about sewage spill in New York State. And here they're talking about the raw sewage bubbling up through the manhole covers and then pouring out into the streets and then going into the coastal water. And they have so many spills in New York State. They actually have a website where you can go and they tell you exactly where the most recent one was and how much it was and how long it flowed for. And you can see the red dots there, they're pretty much everywhere. And they're flowing out along the coast. So the natural route for that sewage is straight into the sea. Now, in that list that I showed from the Damas work, the susceptible whales, minke, there's two species of minke whale that actually appear. And minke whale, there's a population that live off the east coast, the eastern seaboard of the US, and then they annually migrate up to waters up towards the UK, past the UK and into the Arctic. And so if they're filtering large amounts of water to catch their prey and the water is contaminated, then this means that there's a potential for them, an oral fecal route for the virus as well. So clearly at this point, we're now quite concerned that clearly there's an impact for humans, but also for the natural environment and the animals that live there. Now, it was at this point that we started to see news articles about whales. And these are ones in the UK. So this is a fin whale which washed up and died on the UK coastline in February 2020. We had another one washed up on the east coast of the UK. This is another fin whale. And we're starting to think, well, are they linked? Maybe there is an increase in whale sightings off the UK coastline anyway, when that's been occurring irrespective of the pandemic. But this last one was one which really caught our eye. This was a minke whale which was washed up on the south coast of the UK. It's a near adult-sized minke whale. And if you calculate the time it takes for, say, a minke whale feeding off the east coast of the US, it's about the right time for them to migrate up past the UK and into the Arctic waters. And the time it takes for them to migrate across the Atlantic, if you take the average speed of a minke whale combined with the speed of the Gulf Stream, which is the main current, the Atlantic drift, which would take them in that direction. And then the point at which they arrive at the UK coastline is around 10 days, which is also coincides the time it takes to incubate the virus once it's been transmitted. So now we had the point where not only could we had a working hypothesis of how a whale might be infected, and the fact we knew that they are susceptible to the virus, but also there was a clear a link between the two coasts, which was actually made sense in terms of the transition time, but also the incubation time. So now we were, this is the point where we really have to get on with this work and actually investigate this in more detail. So we took a standard chemical pollution analysis approach. It's called a down the drain analysis. It's really designed for identifying what the impact is of people pouring chemicals down the drain, or for example, washing cosmetics down the drains, and then working out how that then gets diluted and pushed into the natural environment. It was originally developed for domestic point sources, but it was then extended to be studied for a countrywide analysis. And all of the data we needed were actually easily accessible because of the pandemic. All of the population infection counts, all of the information we needed for the calculation was all available online on a daily basis, being updated. So it was all easy to get hold of. It turned out that this approach is not new. Using this kind of approach for viruses has been studied before. And this is a piece of work, the Vorma-Marti was work that they published in Nature, the journal Nature in 2010. And indeed, they use exactly the same kind of approach for studying the impact on water security during a hypothesized pandemic. So clearly, the approach we were using was quite valid. But we had to extend it for viruses because the original pollution analysis just meant for an inert chemical. So we had to somehow quantify the viral load that might be appearing in the sewage within the catchment that it's being released in. We also have to know the viable to unviable virus ratios. So just because you detect the virus in the water system, it doesn't mean it's actually sort of an alive virus and therefore able to infect someone. We also had to do a specific uncertainty analysis through the calculation so we had some idea of the accuracy of our approach. And the ones you highlighted in blue, all of those 3 points we could cover from the literature and using standard error propagation, uncertainty propagation approaches, that was fine. But the last issue we had to think about was clearly the virus is likely to die and degrade as it goes through the system. How quickly does that actually happen? We know from previous work that temperature is a strong controller of coronavirus life sort of survivability. But we didn't have any specific data on how this might actually be the case for SARS-CoV-2. Luckily, there was we turned to the medical studies. And this paper by Chin and others that was published in 2020, they looked at the survivability of viruses, SARS-CoV-2 on different surfaces. So there were particularly interesting things like how it survives on counter worktops, chopping boards, masks, and so on. But as a part of that, they actually had to characterize the temperature dependence of the virus at multiple temperatures, and also they had to look at the impact of pH because the virus was being held in solution. Turns out pH doesn't really have a big impact on the survivability of the virus, but temperature does have is a strong controller, which is consistent with what we knew about other coronaviruses. So all we did was we took those medical data and we parameterized, we did an empirical fit to all of those data points. And this allowed us to characterize the die-off, the survivability of the virus with temperature and time. And it was pretty good in terms of, you can see the shaded areas, the tightness of the fit is very, very good. And so this allowed us, we can see here lines plotted at 5 up to 30 degrees temperature. And you can see that the link is, the colder the water, the longer the virus can survive for. So the next problem we had was how on earth do we work out the water temperature of the countries we're interested in? So first of all, to calculate the transmission risk based on the infection rates and dilution from the rivers. But also, how do we then, if we want to work out how that's likely to change over the 24 hours or 48 hours after a spill, we need that temperature data to show that die-off. So for this, we turned to Earth observation and satellite data. And we used an early version of the European Space Agency Lakes Climate Change Initiative dataset. So this was a global dataset of lake temperatures as viewed from a satellite, some 400 kilometers above the surface of the water. But it was a global dataset, and it's down to, it has a spatial resolution of about 5 kilometers. So it was clearly suitable for what we wanted to use it for. So then we wanted to look at the actual dilution. So this is our down the drain approach. And the dilution effect, if you think about the chemical, you pour it down the sink, it would be diluted first by how much water you wash down the sink to get rid of it down your drain. And then if it then lands in the river, it's diluted again based on the river flow and any rainfall going into the river. So the same is true for the sewage. So this means that you end up with a dilution which is country dependent, region dependent, and also both dependent on the domestic water usage, how much water is being poured down the drain to flush that sewage away, but also how much water is in the riverine system and how much it rains in those countries. And so it's not entirely obvious, first of all, how it changes geographically across different regions. And this is a map showing that geographical spread. So if, for example, you can see there's lower dilution in Northern African countries, whereas in places like Canada or South America, then it's very high dilution because they have a high level of rainfall, but they also use a large amount of domestic water. So we had our collective approach. We had all of our inputs. We wanted to try and understand how accurate we think our methods would be or are. Now, we know that the original method was validated for a catchment level, the original approach for the chemical down the drain process. So we had that to fall back onto. We managed to find one data point during the actual pandemic period where someone had measured the wastewater viral counts in Paris on April the 9th. And the measurement they came up with, the 3 × 10⁶ genome copies per litre, was the same order of magnitude as what we would estimate for our countrywide. So we're effectively comparing the results from a large city to the results that we estimate for the whole of France, for the whole country. And so that gave us confidence, really, that what we were doing was valid. That result was also consistent with what we found through our uncertainty analysis. So we analytically identified how accurate our method was. And the accuracy of that turned out to be about plus or minus 68%, which is consistent with that initial result we had from the Paris work. And that itself is also consistent with the analysis of the original method. So internally, this process sort of internally verified that our method was valid. So these are the results. This is the country comparative risk, transmission risk associated with a sewage spill for 39 different countries across the globe. So you can see the plot across the top and then the geographical spread of the risk for the different countries at the bottom. So this is dependent on the domestic water usage and the riverine and rainfall flows within each country. So you can see, for example, the countries, Morocco, so these are the country codes across the bottom here. So Morocco is this first one. This has the highest risk because it has a very low rainfall and very low domestic water usage, versus a country like Canada, which is right at the bottom here, which has a very low risk because they have very high domestic water usage and combined with a high rainfall. So once you have those and you know your infection rates and the number of people in your population, you can then calculate the specific risk associated with a sewage spill, if of course a sewage spill actually occurs through within the country, which is what we've got here. So this is we've analysed this for 21 countries because we only had 21 countries where we could get access to the water temperature data, and you'll see why in a second. And you can see here, so this is Spain first and UK, the GBR is UK. And this is for a specific date for the 3rd of May. So you can see on the 3rd of May, 2020, the highest risk is within Spain and the UK and Morocco. And then it drops away as you go down towards Canada, which has the lowest risk here. Now there's 3 different coloured bars here. This is because we don't know where the viable to unviable ratio of virus is highly uncertain, and therefore we've tried 3 different ratios, so we have a good idea of the potential range in the numbers of viable virus within the water. But to understand these, you also need to take into account the water temperature, the dilution, and also the prevalence of the virus within the community, the country itself. So you can see at the beginning here, Spain and the UK, we have a very high prevalence of the virus. You can see on the bottom plot here. And this is combined with a very low water temperatures and relatively low dilution, which is why the UK and Spain has got the highest risk. Whereas if you go right down to Canada at the end here, they've got a relatively, in comparison, much lower prevalence. But they have a much higher dilution factor, which is why it's much lower risk. You can then look at those results geographically. So you can see the spread. The countries in yellow are the ones where if a sewage spill had occurred on the 3rd of May, then the risk would be much higher than it is, the transmission risk is much higher than it is in the countries where you can see labelled as green, such as Canada. It also means from this analysis, you can actually work out, have an estimate of the concentration, the dose per 100 millilitres of water that actually exists within the river where the spill has actually occurred. So here we have the results for just Spain, the UK and Morocco, again for the 3rd of May. And these are quantities of around 633 copies or cells per 100 millilitres of water. The range itself is set by the fact we're unsure about this viable to unviable ratio of viruses in the water. But to put this into context, 100 milliliters is about one mouthful. So this dosage in Spain it's very large. This is 633 potentially copies of virus in just in one mouthful of water. But you need to remember, or we should remember, that ingesting water is potentially a lower risk than inhaling aerosols or water droplets, because the water has only got one chance to interact with your ACE2 receptors because it goes straight down into your stomach. Whereas if you're inhaling aerosols, then the particles actually go past the ACE2 receptors into your lungs, where there's many more of them, and they're going backwards and forwards as you breathe. So the risk is much higher. But from this, we can also understand how it's likely to change over time. So we can see, for example, in Spain, it has a very high dose to begin with, but 24 hours afterwards, because it's relatively warm water, there's only actually 67% of the original viral load still remaining, and then some 48 hours later, it's dropped below 50. Whereas in Morocco, much hotter air temperatures and water temperatures. And so even after only 24 hours, there's only 38% of the original viral load still surviving. So the important thing here to remember here is that because of the fact that aerosols and water droplets are considered the main route of transmission, there's much higher risk of being exposed to the aerosols that are generated from contaminated water systems like this. So for example, if you have a sewage spill that's upstream of a waterfall, there's a much higher risk of you inhaling the aerosols, the infected aerosols from the waterfall, than there is from you taking a small gulp of the water itself. So these approaches mean that the approach can actually be used in two different ways. So the method I've described can be used in a forward calculation approach, which is what I've described, the order in which I've described it within this analysis. So effectively, it means you can try and identify the potential transmission risk of a particular water system to advise the population, stay away because there's been a sewage spill and there's a high risk of the virus becoming airborne. But it also enables you to identify possible locations where water transition may occur. So it means that you could feed this information into a track and trace method. So current track and trace methods only consider person-to-person transmission. They don't currently consider the potential of the environment itself to transmit the virus. And so this kind of approach could provide data for those approaches. It would also allow you to identify potential animal populations that have been exposed to the virus, such as the whales we were talking about at the beginning, but also other animals that may be living alongside riverine systems, such as ferrets, or which potentially could become animal reservoirs for the virus itself. The other way it can be used is in essentially a backwards calculation. I talked at the beginning about the fact we're uncertain. This ratio of the percentage of viable to unviable virus is highly variable, and at the moment there's a high amount of uncertainty around that ratio. The methods I've described here can actually help you go back and reinterpret those data. And it shows that to be able to identify whether the viable virus is appearing in the natural environment, you have to understand the complete temperature history and the age of the sample itself. So you have to be able to quantify how long it was since it left the person's body and became sewage to the point where you actually sampled it. And only if you understand that complete history of the sample can you then interpret any results you have from your actual sample analysis. And so it's possible that the conclusions of this work not only will help to improve and reduce the uncertainties of the approach itself, but it will also prove to be useful for wastewater epidemiology approaches. So that study that I talked about at the beginning that initiated and started our interest in this whole piece of work would itself benefit from the results of our analysis. So everything I've talked about so far has been a country-specific or large regions sort of state analysis. We haven't talked about ultraviolet light and the impact, other methods that may cause degradation of the virus. We did look into the impact of ultraviolet light, and it turns out that the most damaging part of the spectrum, in terms of for the virus, is actually the part of the spectrum that's actually absorbed most by the water. So it's unlikely that ultraviolet light will cause any major degradation of the virus. But we haven't looked at biological or bacterial influences, or even the deactivation of detergent, because often when sewage is washed down a system, there will be detergent present as well. But if you did want to apply this to a catchment level, it's entirely possible. The original method was validated for and developed for a catchment level analysis. You just need some catchment level input data, so things like population infection rates, domestic water usage, river flows, etc., for your actual catchment of interest. You need your water temperature data, and the satellite observations we've used for this analysis were, they're at 5 kilometre spatial resolution, and so they're completely usable for catchment level analysis. But the biggest hurdle you'd have to overcome is the fact that the dilution factors, which we took from the literature, are likely to be highly variable, temporally variable and spatially variable within some countries. So you'd have to have some sort of catchment level, higher spatial resolution analysis for those. Now, before I finish, I just wanted to touch on the publishing experience we had with this piece of work. I described at the beginning the kind of expertise we had within the team. And so we had people that covered everything from water quality, maths, modeling, uncertainty analysis, right through to epidemiology. And that was key for us to be able to do this work. But the work was actually passed through 3 different journals and 8 reviews before it was actually accepted for publication. And as a part of this process, we experienced quite a lot of reviewer resistance and dismissive statements about our work, and some reluctance in some reviewers to even consider our work. And the conclusion we came to on this was because we were new to the area of viral epidemiology, rather than not new to the area of water quality and sensor and understanding and modeling and uncertainty and all the other components that are involved in the work. We also, over the same time, we had 2 proposals rejected, and again we had the same experience with the reviewer resistance, and kind of it was like people gatekeeping effectively the area of research and the resistance to new people being involved. And in the end, it was actually advice that was provided by a professor at Princeton, one of the editors of the journals, that allowed us to identify what it was that they didn't like about our piece of work, and then enabled us to address it. But I don't think this kind of experience is really limited to just virus research, and so I wanted to highlight it because I don't think it's unique. And it really highlights the need for the importance for interdisciplinary work, but it also highlights the barriers that you can actually come upon when you try and publish and use and develop this kind of approaches. But I'm happy to say the work is now published, and it clearly was very well reviewed. We had a lot of reviews, and currently has been read over 6,000 times, and the work's already been cited more than 30 times, and it hasn't even been a year since it was published. All of the tools I've talked about are all freely available. All of the software there's a spreadsheet you can use to do the calculation of the risk, and all of the Python utilities we use for the paper, including the generation of the plots, is all freely available on GitHub. And as is the paper itself, so the work is published in Environmental Science and Technology, Water. It's open access, so it's free to download. And there's the link at the bottom that gives you access to all of the tools that are on GitHub. So thank you very much for listening, and I'll be very happy to answer any questions that you may have.
Dear Jamie, thank you very much for this excellent presentation, and also for making your tools available, openly available at the GitHub. And I'm open for questions from panelists and audience, please. And just for general information, the questions will be registered and the answers can be later prepared or answered by Jamie and posted on the Air Center website if required.
if there are no hands up, may I start it off?
Of course.
Jimmy, thank you. That was a fascinating talk. And really, it highlights why we need something like the ONWARD network to bring all the expertise together, along with the medical community, the practitioners, and the general public, to make this kind of work effective. Otherwise, as you say, it may not get published. If it does get published, it might sit somewhere inside the journal and nobody benefits from it. So really appreciate this talk that you have given. I think you touched upon it, but may I ask how easy it would be to also to adapt the model to other types of waterborne diseases?
Yeah, sure. I think it'd be relatively simple because it's, as long as you have the set of the equivalent set of virus information about its about the survivability, the ratio of genomes for a particular virus, then it should be applicable to all types. And certainly the work I highlighted at the beginning that was published in Nature, they used the same approach across multiple virus types for a global analysis. So it wasn't, the original work wasn't a global analysis, wasn't applied to coronaviruses. It was, so I think it would be highly applicable.
Yeah, so that'd be great, because this, I mean, in all the problems that came to the fore because of COVID, there are many other forgotten pandemics that we need to start looking at as well. Yes,
absolutely. And a lot, for example, a lot of our early work was actually informed by the MERS pandemics, as well as the SARS-CoV-1. And so we built upon, and it was quite easy to see, and you could track this progress of people using other diseases and viruses to then inform how they then approached with SARS-CoV-2.
I'll stop because I see that Grinson has his hand up.
Yes, please, Grinson, go ahead.
There is another question also in the question box, 2 questions. So I will also, this question would have been nice to be addressed to Tom, because I was listening, while listening to you, I knew that Tom has developed something on malaria epidemiology, which will be an interesting thing to look at. Can you share that link also? Because we are looking on something similar in our waters too.
Yes, I'll be very happy to. Yeah, very happy to give you Tom's publications. Yeah.
Maybe we should invite Tom to give another webinar.
That's true.
He's now based in Germany.
From the audience. Jamie, Joana Soares, she was asking and mentioning, she was thinking that there are several chemical compounds, for example, endocrine disruptors that are not completely eliminated in water treatment plants. In the case of virus, she would be presuming that UV radiation and detergents used at the facility could eliminate the virus, but what is failing, or moreover, what can be improved at wastewater treatment procedures?
Yes, so that's right. If the water is completely processed, then it will kill the virus. And so the United Nations and the World Health Authority guidelines for purifying water are completely, they work, and they are the sort of, for example, the use of chlorine to deactivate the virus, it is sufficient to kill it. The problem comes when either the equipment is not, those guidelines are not being applied correctly, or if the sewage spill occurs before the water is actually being treated. So this is, really, the work we've talked about is really considering raw sewage, so this is untreated waste. It's been collected but hasn't had any treatment of any sort. And so the first the water treatment will tend to have different stages. The first one will typically be some sort of sediment settling process. And that settling process itself would take a lot of the virus out because the virus attaches to things that are in the water. And so when they settle out, it goes with them. And so as you go through all the different stages of the water processing system, the amount of virus will naturally reduce, and to the point where once it's been applied with the UV system over a very small area of water, an intense amount of UV, or using chlorinated method, then that's the point where the virus will be killed, and so it won't be present in the processed water that comes out at the other end.
We have another question coming from Vivian Silva, and if she's asking if the transmission risk have been calculated for dates after 3rd May 2020, or it would show significant differences if it were calculated during different times of the confirmed cases curve, for example, maximum and minimum cases and so on.
Yes, we haven't, we didn't calculate it for other dates. I think we used a couple of example dates just to demonstrate it, but we weren't able to. We actually applied for one of those funding proposals we applied for was to fund us to actually carry on and actually plot them for all of the countries we were studying through time, so that we could actually provide that as guidance to the policy. But unfortunately, we weren't funded to do that, and so we weren't able to do that. And it will change quite a lot. So the risk, the transmission risk, will change based on the number of infections within the country and the environmental conditions. So as the seasons change, you get differing amounts of rainfall and river flows, and so the risk will change probably on a month-by-month basis more likely than day by day, but they will change through time.
Any other question or comment from the panelists?
Yes, Jamie. Good evening. It was an exciting talk. That's a new topic, and viruses in the wastewater treatment systems and possibilities. I missed some part in the initial say because my internet was not good. So somewhere where you discussed about viable viral load, I missed that part, how you consider the viable viral load. And second thing is, my question is, how adaptable this model or this work for a, area like in where the non-point sources of pollution is much higher, and do you consider the population density? Because when it comes to the catchment areas, dilution factor is different, but I understand the population density is low. But when it comes to the coastal cities, then we have more factors. One is the population density high, then non-point sources of pollution is high. Then again, as you said, the limitations of salinity, which we didn't consider, that also will come. So it becomes a more complicated system, right? So can you give some idea into that?
Yeah, okay. So the first point you raised was about the viable to unviable ratios. The reason for that is because if you detect the viral RNA, the acid in the sample, it doesn't necessarily mean the virus itself is alive. It might be that it's just dead and it's the remains that you detect. So you have to be able to quantify how much of the detected virus within the sewage is actually viable or infectious virus. And there's been lots of sampling of that, and there's been mixed results. Some people have not detected any viable virus, some people have detected viable virus, and the ratios have varied. And part of the reason because of that, or part of the reason for that is that we think that you have to know the temporal history of your sample before you can interpret your sample results. And often, because of dilution, the sample quantity is likely to be below the detectable limit of the instrumentation which is used to detect it. And so that's why we have no clear idea of what the viable to unviable ratio is. So what we did was we took some published literature, this ratio for adrenoviruses. And we took 3 reasonable ranges which went from 10%, 1%, and then 0.1%. And which is what other studies have used for SARS-CoV-2, so we were consistent with other work. But the idea there is it gives you kind of a lower and upper range of the quantities that you may expect to find as a result of this, the uncertainty in this ratio of viable to unviable viral copies. And then your second point about the near coast environment, and the fact it's much more complex, and there's the pressures are likely to be different at the coast versus higher up in the catchment. Yeah, so all of this work we've done is taking kind of the average approach. We've assumed that you have a large population somewhere in the catchment, and we've actually used a log because of the impact of super spreaders, where you potentially have one or two individuals which may actually contribute the majority of the viral load which you have within the water system. The approach actually uses a log normal distribution to account for that. And so we just assume, the approach assumes that somewhere within that catchment, there is at least one super spreader. So the fact you have less people in the upper catchment versus lots of people at the coast, it just adds weight to the fact that somewhere in amongst that group, you're likely to have a super spreader. And so we're very much looking for we're modeling or estimating, calculating the average result across the whole catchment. But I think if you wanted to apply it at a catchment level, you really need to have some, as you say, some idea of how this dilution factor varies within your catchment. Because it is, in some countries, particularly some parts of the UK, some parts of Europe and Asia, you will have some countries that have large variability in this dilution factor, and so that is something that can be quite important. Whereas, if you're considering countries like Morocco, for example, they'll have a much tighter constraint on the dilution.
Thank you. Any other comment or question? So if not, I would like to thank everyone for the participation, and again to Jamie and to the Air Center staff. We'll continue with the ONWARD webinars during the next month. Expect some exciting mornings or afternoons or evenings. And the same for the Air Center Networking Fridays. For more information about this and previous sessions, please visit the Air Center website. Thanks you all. Obrigado. Bye-bye. Shuba, would you have any words, or José, maybe?
Only to say thank you to you, Milton, for running the show. And many thanks to Jamie for a really fascinating talk, and to all the participants for your interest. But I also want to make a plea for all those who are in the audience to please subscribe and become a member of the ONWARD network. José has been putting in the link to the email address to which you just have to write and say, I would like to become a member. It's important because this is a network project, and only if you join, we know that you are interested in the problem. And as Jamie's work has illustrated very clearly, we need expertise in different subjects and different backgrounds to come together to address this problem effectively. And as part of the ONWARD's network activities, we are building a website, and we would like to develop it as a resource for all the interested people. For example, if we get a list of your publications that are relevant to this problem, we would like to make it available, or at least make the links available through the website and so on. But this initiative will only become effective if more and more of you become actively engaged in the network. So please consider that. I wanted to make that pitch. And many thanks to José for having us all become part of the Air Center webinar activities. I think it's been a great help and a great success. And thank you all, panelists, participants, all of you. Thank you very much.
Thank you.
Okay. Thank you much, Shubha, Milton, Jamie, fantastic presentation. Bye, Grinson.
Thank you. Bye-bye.
Okay,
Thank you.
bye,
Bye.
bye, bye. See you soon.
Thank you.
Thanks very
Thank you,
much.
Jamie.
Bye, bye.
Thank you, Jimmy.
No problem.